Welcome to Under the Microscope, the biology podcast by Cheryl and Sophia. Join us in our cozy biology corner and get ready to smile, laugh, and learn about the amazing scientists doing good and changing the world through their research. We have long-form interviews of researchers, scientists, and professors with topics from pandemic prevention to cancer cures all the way to genetics. Or you can join us for a short snippet of a bio bite. Be kind, lovely, and stay curious. Hello, curious people. Welcome to Under the Microscope, where we are exploring the biology research making the world a better place. Join us as we discuss articles, papers, and interview experts to explore the cutting edge of biological research, or take a quick bio bite to go about new scientific developments. Today, I had the pleasure to speak to Professor Greger Larson, who is the director of the Paleogenomics and Bioarchaeology Research Network at the University of Oxford. From why the word domestication isn't as clear-cut as it seems, to whether we should all be sequencing our dog's DNA, and all the way to how chickens bring together evolution, genetics, and a lot of very interesting human history. I can't wait to share his wonderful insights with you in this episode, Season 2, Episode 1, Uncovering Our Past, Domesticated Animals to Human Development, How Bioarchaeology Reveals Our Evolutionary Origins. Hello, thank you very much for speaking to us today. How are you?
I'm good. Thank you for having me.
I'm very, very grateful for your time, and I'm sure many viewers are very excited to listen to you talk about genomics and evolution and bioarchaeology. Could you tell us a little bit about what your current role looks like and what you do in your day-to-day?
Day to day, that's a great question. I run a lab. So I am based in the School of Archaeology at the University of Oxford, and I run the Ancient DNA Facility, which we call the Paleogenomics Research, and so the Paleo Barn, which is a backronym. And what we do is we sequence DNA from mostly things that are dead, occasionally things that are alive, or at least were recently alive. And we get those sequences and we compare those sequences to other populations of both dead and alive organisms, mostly animals, occasional plants we will play with, and all very much in service of just trying to understand the relationship between people and animals in both the recent past as well as deep time, as a way just to try and understand our on this planet and how all of this stuff came to be, and the ways in which the whole myriad of evolutionary and cultural history have kind of intertwined to create the modern world.
We'll touch on the techniques that you use to be able to extract this DNA and make inferences. But for now, could you please tell us a little bit more about paleogenomics, about how the findings that you make could influence our understanding of the past and the current, and especially how DNA evidence can also give us understanding about behaviors, cultures, societal development?
Yeah, that's ultimately what we're after. It's not straightforward, but it's always a lot of fun, and we try and bring a lot of curiosity to the kinds of data sets that we generate. We should also be mentioned that very often what we do, and we always endeavor to not just rely on genomics, but to incorporate a whole bunch of not just the context of where the samples that we get to work on come from, but then other kinds of evidence that you can glean from that, from stable isotopes to radiocarbon dating to morphological analyses like geometric morphometrics. And very often those techniques in combination with the kinds of samples that we look at and the kinds of analyses that we use lead to even deeper insights, which is revealing of that kind of long-term nature of the relationships that people have had with animals, going very far back in time. And one of the reasons that it's so much fun for us is very up until maybe 30 years ago, if you were looking at bones in the past, you were really relatively restricted into what you could say because you were relying exclusively on the morphological shapes or the patterns or the number of bones in relation to other things. And you could identify a lot of things to species. You could identify butchery patterns and cut marks and all kinds of other things. sex ratios. There's a whole range of different things that you can infer by just looking at zooarchaeological assemblages or paleontological assemblages. But what you didn't have access to was a much finer resolution of DNA. And that has really only come about in the last 30, 40 years, where you now have access to, for some samples anyway, the DNA held within them, which again gives you this real power because you have the resolution, the resolving power of a very fine scale differences, but lots and lots and lots of data points. I mean, you and I have over 3 billion base pairs in our genomes, which is a huge number. And a lot of those vary. And that means we can look at that variability between not just human populations, but animal populations, plant populations. And then if we can do that through time, that gives us a continuum of change. So rather than having to just simply infer the past from the present, we can directly look at it and we can compare those populations in the past with what they are now and look at those patterns of selection or of admixture, of relaxation, of all kinds of different things to see how those different animal populations responded to the different circumstances in which they were living with people or outside of people's experiences and pressures, and how that whole thing has come to shape the modern world, because so much of things have changed in just the last, say, 50 years or the last couple hundred years, so that trying to divine much beyond that from modern populations becomes very fuzzy very quickly. And so having access to the past with that degree of resolution just gives us a power that's, for us is extremely exciting because during my PhD, I focused a whole lot on an 80 base pair stretch of pig DNA. And so 80 was great. 80 was a big number. And a lot of those positions varied, and I was able to make a lot of them. But just recently, we generated, was it like 20 billion DNA from just a single individual? And we're dealing with hundreds of individuals. And so to go from 80 to multi-billion base pairs from any one individual just shows just how remarkable it is that the change that's happened within the field and the kinds of power that we have now to infer the past.
And of course, the field is so dynamic with differences in technique and the instruments, but how exactly did you get into this position? What drew you from your PhD to where you are now to continue exploring bioarchaeology?
It's interesting that I'm actually doing the same thing now that I was doing for my PhD, which I'm just using new, better tools with greater powers of resolution. What got me into the PhD in the first place was I've always been just really curious about. I just was always really interested in evolutionary biology. I just love this idea of change through time, of the different ways in which organisms interact and how that changes the reproductive, who lives and who dies through time and space, and how that might then manifest in populations and looking at them change through time and space, both in their terms of their distributions as well as their genetic and morphological constitutions. And I just always thought that was really interesting. And I also then really like the idea of looking at something where it wasn't just the individual species on their own, but that through, if you would put people into it as well, now you have people plus animals. And that then adds a cultural dimension to it. So then by combining the cultural element of human animal interactions plus the biological side of human and animal evolution, for me, that was just this amazing kind of like, wow, look at what we can do with all of this. And that meant that we could start to address a lot of the questions around origins of, say, agriculture and different ways in which humans and animals have both coexisted and have kind of benefited from each other's close presence of each other, and just trying to work out, what happened over the last 10,000 years. And, I'm not sure that anybody really knows some of the larger questions, like how much of this was deliberate and why didn't this happen in 300,000 years prior to this when we were a very good species and looked just like us now? But something was clearly changing. And I was just really curious to get to the heart of that. And I felt like one way to do that was to be able to bring, again, that kind of resolving and super high powered genetic technique, but using that on the past. And I thought that would be a really fun window to try and investigate all these really big questions.
And of course, it's very unanswered and interconnected and broad, but do you have some favorite case studies of inferences we've managed to make in either animals, humans, or our relationship together through paleogenomics?
Yeah, so one that I really love, it's been a couple of years since we published this now, but I'll tell you why I love this for a variety of reasons. That there was a gene called thyroid stimulating hormone receptor, which is found in lots of different things. And it was identified just over a decade ago, I think, in chickens as having been fixed in modern populations for one type, and they called that sort of the domestic type. And then when you look at red jungle fowl, it seemed to be fixed for the wild type. So it looked as though every domestic chicken, and of which there's now multi tens of billions, possibly 80 billion, certainly the most numerous domestic animal on Earth, and by weight, the most numerous bird on Earth as well. So we've just taken chickens, just proliferated them like crazy. And every single one of them has 2 copies of this derived allele, this derived mutation for TSHR. And the wild one seemed to be 0. So that suggested to the people who had written this up that, it's a big result. And it's like, well, maybe it's a domestication gene. Maybe it's something that was absolutely critical at that shift between wild to domestic. And maybe that happened a long time ago. And that was absolutely essential for chickens becoming what they are, and how they then follow this trajectory away from their wild ancestor to something that was more amenable to living in such close proximity to each other, proximity to us, and being able to reproduce in those circumstances as well. So by getting some ancient DNA from some ancient chickens going back a couple thousand years in Europe, what we were able to show was that for the first, after chickens arrived in Europe, which is only 2,500 years ago or so, for the first 1,500 years, you would expect that by the time they get to Europe, because of course they're domesticated in Southeast Asia, that they would be fixed by then too. But the first populations that came in, it was only at 40%. So we were like, well, that's kind of interesting. So when was this selection pressure that allowed it to go from 40% up to 100% and fixed ubiquity in all modern populations? And by using some genetic techniques associated with applying models to selection pressure to see when all this might be happening, it looked like it was about 1000 AD when the selection pressure really started to rise above 40%. And then it got fixed only in the last 100 or 150 years or so. And it was like, wow, okay, so far from being a domestication gene that was absolutely essential at the very beginning of the process, it looks like that fixation was really relatively recent. But that was only the tip of the iceberg, because then when we started looking at other aspects of this, when we look at the zooarchaeological evidence, it shows that prior to 1000 AD, chickens were about 6% or so of the total number of animals, or the proportion of animals within any archaeological site in the continent and in Britain, from say 0 AD to 1000 AD. But after 1000 AD, it almost doubles, and it gets up very quickly to about 12%. So there's clearly an increase right about 1000 AD, which is precisely when the genetic evidence is telling us that there's a selection. And at the very same time, we started talking to some historians, and they're like, oh, it's interesting because a lot of governments at that time started really heavily enforcing this idea that over fasting periods, which at the time was more than a third of the calendar in Europe, that you were not allowed to eat any 4-legged animal, but 2-legged animals were okay. And now we have both a genetic signature of an increase in selection pressure for a gene that allows a greater proliferation of chickens living in closer proximity to each other, with that very clear demonstration across a wide range of zooarchaeological sites over 2,000 years in Europe, showing a significant increase in the proportion of chickens relative to all other domestic animals within archaeological assemblages. And then we have the politics and history showing the kind of motivation for all of this, which is people like, oh, well, we can't eat the 4-legged animals for a large proportion of the year. Maybe these chickens, which had been consumed at this point, but relatively small. I mean, 6%, it's now like the single most consumed meat, but at the time it was really, really minor. And for a while, when chickens first arrive in Europe, nobody's eating them at all. They're kind of revered and they're buried with people, and they're protected in a way. So by combining all these different lines of evidence, not only did we demonstrate that this gene was far from kind of a domestication gene that was important at the beginning, but that it followed this really, as you said, dynamic relationship with people, whereby you have politics driving behaviors, the end result of which is an increase in selection, which fixes a gene, which then contributes to a significant increase in the number of chicken bones that we find in archaeological sites. And they're all pointing in the same direction, which is 1000 AD, and all of those independently as well. None of them we were relying on any other data sets to do that. And so for me, that was just a brilliant study that showed the power of not just the increased resolution of being able to infer selection at a genetic and biological and evolutionary perspective, but then combining it with a whole series of other very well-established methodologies and very disparate fields that were then all very much pointing in one direction, which is like, okay, something is clearly happening at 1000 AD with chickens, and that yields now where we are today. But there's all kinds of change that have happened since then as well, but that seemed to be a watershed moment in the real significant change in the relationship between people and chickens.
If anything, there seem to be more questions now than answers. But in terms of the whole length of this particular study around the genes, are there any bottlenecks at the moment in finding the evidence or acquiring the ancient DNA that aren't to do with precision?
That's true. I mean, in any samples are going to have questions about provenance, about permissions, about ethics. And I'm really pleased to have seen that, and certainly in my time in academia, that's really come out in a significant way. And it was only 20 years ago it was possible to travel to lots of places and just pick up the occasional feather or a bit of hair or something and bring it back to the lab and analyze it. And now, of course, we know much better than that. And it's now about recognizing that these things are these living, breathing animals. Firstly, there's the animal on one side, so you don't make sure that you're not harming this animal in any way. But then there's also this is a resource for that place that you're in, and so you need to be taking that into consideration. Having local partners who are aware of the kinds of analyses that are being done and what it means for them, and the kinds of questions you're asking from the beginning, because you don't just want to from the hip and say, oh, we're just going to grab everything and sequence everything and ask questions later. I mean, there's always an element of that in science, of course, and there's certainly a place for that. But it's nice to be able to say, we have this specific question that these particular samples are really going to help us address. And here's the context for all of that. And here's what we would propose. And how would you inform what we're doing as well? And so it all becomes a very cross collaborative kind of win-win for everybody. You're considering these things. And then, of course, we 15 years ago or so, 10 years ago, we spent a lot of time in Russia collecting samples with a lot of people on the ground there. And of course, since now the war, that's become very, very hard to do. And there's been a lot of, if not impossible, and a lot of kind of people looking at our grants and saying, oh, do you have Russian partners and all these things? So you're trying to do science and politics is invading, and it's like, okay, well, you can't just ignore it. Like, you have to address it. You have to be a part of it. And for us, we try and be ahead of it, and we try and ensure that we're starting off with the right idea, but certainly open to new ways and other ways of doing things, and negotiating and talking with people about what is the best way forward for everybody.
And I really loved that you touched upon the idea we come in with questions first. Do you think that as a field as a whole, that there are some very prominent questions that are actively trying to be answered at the moment?
Oh, absolutely. I mean, we are never short of questions. And as you've just said too, like, very often the answers that you get only ask more questions, right? Like, so, in the case of the chickens, yes, okay, 1000 AD in Europe, but chickens arrive very late in Europe. They're only, it's 1000 years, 1500 years or so after we get the first chickens ever in Peninsula Southeast Asia. Is there any evidence for people eating them in South Asia or East Asia as they're making their way across Eurasia? You're like, what are the religious implications of what's going on through there? How are those chickens being integrated into human societies before they reach Europe? What's happening to those genes on the ground in those places before? I mean, how did it get from 0 to 40%? Is that having to do with the subspecies of chickens in Peninsula Southeast Asia? Or is this really a selection pressure that's getting up to 40% where it sticks there for a while and then goes to 100%? I mean, we don't have answers to any of these things, and yet we wouldn't have known to ask them unless we had that initial data set. So every new result generates a series of questions like, oh, wow, we really want to get to the bottom of this. We need to know that. And then you do that same thing. You go, oh, now that we know that, we need to know this. I'm pleased, though, that for a wide variety of taxes that we're working on, I feel like we've made a real contribution to our overall understanding of its long-term relationship with people. And you could say domestication for that. I have issues now with that word, but just because I think of how easily it's misinterpreted and what people often think what it means when it doesn't, and it just gets very complicated very quickly. But so there's elements of knowing all of that, which I think we certainly know a lot more than we ever did, but the amount that we don't know still far outstrips what we do.
And I'd love to hear if you could break down the main arguments for and against the word of domestication, because it's obviously quite an abstract concept, and biological domestication isn't an on and off allele.
Yeah, I think the issue with it is that it's a kind of a preloaded term. Like, it's one word, domestic or domestication or domesticated, and it's one word, but if you just kind of peer behind it, you're going to see an entire compendium in people's heads of what that actually means. And usually that means some sort of intentionality, some sort of control, some sort of tameness, all these kinds of manipulation, or we're doing it for our benefit. And it's kind of there's a real rigidity to the way in which people use that word, and its definition lacks a kind of applied specificity. Like, it's not it's so loaded with all this baggage that I feel like every time I use it, I already know the way in which people are misconceiving what I'm saying. So much so that I don't, even earlier when I said chickens were domesticated in Peninsula Southeast Asia, I know how people are going to take that. And I don't mean that, but for a shorthand, I will say that just to try and get away with it. Because as soon as you say something was domesticated, or to domesticate, it's a transitive verb. It's something that happened. You have the person doing it and the person receiving it. And that's not how I think the actual process works. And so embedded within the term is an assumption about the process. And I think the way that we perceive that process, frankly, is completely wrong. for a lot of reasons to the way in which we perceive the whole natural world, which is that there is very linear development from point A to point B. It's all very deliberate, and this is how things work. And we have the same issue with an evolutionary biology. We personify animals, we give them agency, we give genes agency, we do all this, and we do that because we're storytellers, and because that's the way we understand things and the way we communicate things, even if we are also aware that at the same time we are wholly misrepresenting the process itself, or at least loading it with baggage that just sinks it down in terms of its ability to be truly comprehended if we really wanted to know the answer.
It sounds a lot like the Five Stages of Man poster with the idea that everything is linear, but is there a particular phrase or a particular way you like to describe a subgroup of domestication in the form of, for example, dogs being domesticated despite it being not static and very different? A way that you wish you could say it for people to interpret the process more correctly?
Yes, I do wish there was such a word, and I have yet to find one. This is why I often I fall back on relationships. And if we're using evolutionary biology, kind of emergence. And so what I like to say often is that if you have a wolf population here and a people population here, it's through their interaction that dogs then emerge. So therefore, you can get away from this. People did this to dogs with the intention of producing this, or yeah, so dogs are therefore an emergent property that arises from a relationship. And what we want to try and figure out are what are the timing, the spaces, and the places, and the kind of interaction that define that relationship that then led to dogs. And that, I think, but you see how wordy that is, and you see how much of a problem it is. Or somebody could just say, you mean you want to know how dogs were domesticated? I was like, well, yes, but no, right? Like, I mean, yes, but without this idea that we did that, which is what dogs domesticated implies. And that's where it becomes problematic, because, and I think it's true of anybody who's researching something, that the terms around it become problematic very quickly, regardless of what it is you do. And I had this thought a little while ago where I could imagine somebody saying, oh, well, Greger, you're being too much of an academic, and your ivory tower, and you're worried about the specificity of the terms. I'm like, well, yeah, that's true. And they're like, I could see somebody going, you're adding nuance where it's not necessary. And I would say, actually, because there's all this baggage, what I'm trying to do is not nuance. I'm trying to get to a more sophisticated perspective, because I feel like you can lack nuance and still be okay, but nobody wants to be unsophisticated, right? I mean, I feel like it's more robust to say we have a much better understanding of the process. And if in order to get there, we have to dispense with some of the terminology which infuses a kind of package deal of how we interpret this, then that's fine with me. But it does make it harder to do in the literature. It makes it harder to do if you're talking with somebody at Thanksgiving, or over Christmas or whatever else. But for the purpose of your podcast, I think your listeners will appreciate that sophistication is not a terrible thing.
It especially reminds me, despite not being genomic, but of looking at our past, of the alpha wolf theory, which was definitely lacking both nuance and sophistication, and the actual scientists behind it definitely had the nuance in mind, but obviously communicating that to the public, the media kind of runs with one interpretation. Do you think there are any bioarchaeology or paleogenomic examples of this sort of sensationalization bias?
Uh, Everything we do, I mean, it's a double-edged sword. One of the reasons that we started doing dogs in the first place was we were working a lot on pigs, and I was relatively early in my career, and I was really had been interested in dogs for a long time. And I saw a lot of papers coming out that I was, without being too negative, I was dissatisfied. I just thought, you could have done that a bit better, or that's not really saying anything. And what it seemed to be pretty clear to me that a lot of these papers were passing peer review at journals where it seemed to me that if they'd been working on another animal, they wouldn't have gotten through. There was, in other words, that the fact that it was dogs meant that the reviewers, the editor, they were aware of clicks and of possible press, and it was sort of the slide for these papers to get through, which we now have absolutely benefited from, right? We've published a bunch of stuff on dogs that I guarantee, if we've been working on Drosophila or Arabidopsis, there's no way that we get either the publication venue or the subsequent press. And so it's very easy for me to go, oh, those dog people are publishing where they shouldn't, get all high and mighty about it. It's like, aren't you benefiting directly from this? Yes, I am, all the time. So I don't want to, I'd be a complete hypocrite to say otherwise. It is not just true of dogs, and not just true of genetics, or certainly ancient genomics, but I would argue all of archaeology. I mean, it's amazing to me. I was just in Pompeii weeks ago for the very first time, and the number of people there is just insane. It's like it goes back and forth between that and the Colosseum is the number one most visited tourist place in all of Italy, and it's fascinating. It's absolutely amazing because you're getting day-to-day life experience, and we are, of course, drawn to stories about ourselves. We're drawn to stories about who we were in the past and our ancestors. I mean, you just look at how many people have thrown their DNA to 23andMe or Ancestry.com or whatever else, and or done the same for their dogs, for that matter, right? We are eminently and kind of unwillfully attracted to these things, which means we're paying attention to them, which means we elevate the importance of them because they mean so much to us personally. And so, but with that, as you've very clearly demonstrated, becomes a kind of stripping of, for lack of a better phrase, that nuance, or for a sensationalist quality, or for people like, what was it, Netflix last year, where just basically conspiracy theory dressed up as archaeology for 10 parts. And it was like, okay, great. Like, as though we don't have robust techniques to figure this stuff out. It's like, oh, all these archaeologists are hiding stuff. It's like, no, we're not. Like, we're simply after the truth. So, um, yeah, I mean, absolutely. We both benefit from it and kind of decry it at the same time because that extra focus and attention means that it's great and it's terrible all wrapped up in one.
Of course, you mentioned 23andMe and Ancestry, and there are many ethical and social and racial issues with how they conduct, and also the cybersecurity of storing all of your DNA online in someone's hands. But what do you think are the benefits if they're to sequence their DNA? Or do you think all of these inferences are pseudoscience and potentially could they be used for any sort of public research? Or do you think that it's a waste of time and money?
It's absolutely, it's not a waste of time and money. I think that's making it much more of a black and white thing. And it's, of course, like any technology, nobody really, internet first came out, everybody was super excited. Nobody thought it would necessarily lead to bubbles of hate where everybody just kind of, and huge amounts of undermining democracy and all kinds of other terrible things that have resulted from the internet. But at the same time, we couldn't live without it either, right? I mean, it's like anything that there are positive and negative sides. And I think that it's just, you need to, it's about educating ourselves about what those potential negative sides are. And as you rightly described, that in fact, there was just an article in the Atlantic recently about like if you gave your money to 23, if you gave your genome to 23andMe, and then they go under, which is a possibility, and then they sell their database onto a different company. Now you click a box and it says, oh, new ethical standard. Now we're going to sell this off to insurance companies. We're going to do a lot of it to the police so they can track down people who've committed crimes, but maybe without your consent. So even, great, we got the bad guy, but so it gets tricky and sketchy in a hurry. But what's absolutely clear is that it's very gray, and it really depends on a case by case basis about whether or not, and things that are great now might not be so great in 10 years and vice versa. So I'm not giving my DNA to anybody for stuff, and I think that a lot of it is pseudoscience around like your dog is 18% Bichon Frise. There's a lot of sketchy stuff around all that, but if it scratches that itch and you want to know, and if it makes people pay more attention to the kinds of research that we're doing, and it gives them more empathy for somebody else's dog, then maybe it's not the worst thing in the world.
I mean, when I see my puppy wolf down her feet, I know she's 100% Labrador, and then once again act as if she is starving. But
But being 100% anything might not be a good thing either, right? Like,
yeah.
we get into essentialist ideas, and frankly, the beginning of dog shows in the UK and in Europe at the beginnings of what then became what we now know as modern breeds, which is how we see dogs now, was just the first step toward a very racist ideology, which was adopted by the Nazi Party. So, you look at your 100% Labrador and you go, I wonder how this could be misused with government applications toward who are the good people and who are the bad people. And it's like, yeah, it gets complicated.
So we always joke that in her DNA is also shark, piranha, and anything else with teeth. But on the topic of technology being a double-edged sword, do you think that people treat science as, I guess, a static database of facts that you kind of disprove and sensationalize rather than science as a process of getting data that we then infer? And do you think that this has been recently seen in, for example, biology as a whole or in the study of genomics?
Yeah, that's a good question. I feel I don't know that scientists have always been the best advocates for science. And it's very easy. And I have some colleagues who have done this, which is that we just put out the data and then we wash our hands of it and then it just gets interpreted. And it's like, it's very easy for things to be misinterpreted. And I think it's important to consider the ways in which your research and your narratives and your conclusions can be misinterpreted, so as to make sure to minimize that likelihood in your papers, in your press releases and whatever else, while at the same time being aware of the biases that are within science itself. I personally have benefited from getting a lot of press, which means that maybe my career has gone places it wouldn't have if I'd been working on something else. So, yeah, there's the kind of here and now benefit versus the longer term benefit versus the personal versus society. I mean, there's all those kinds of things, but I think the more aware of it we are, and I feel too that we need to be talking about science as you refer to it, as a dynamic kind of human driven affair, and with all of its warts and all, right? I mean, there are sometimes that, and we recognizing the way in which we understand the world, which is through narrative, which is great in some cases and terrible in others. The way in which people are, which science has been abused and can be abused, like anything, and but also being aware that while we shouldn't have complete fealty to scientists as some sort of like better than thou anything, the complete rejection of anything to do with expertise is equally terrible. And we have politicians who have said as much, like we just need to get rid of all the experts. I thank you, Michael Gove. So it's like, it just seems to me that there are ways in which we can increase understanding, increase empathy, and increase respect, both for one another, for our present and our past, by being careful with our language and careful about the way in which we communicate the kinds of things that we do and the importance of it, without either hiding behind it or going out all bells and whistles, being like, this is the truth and that. And it's just there's subtlety to it all, and it depends on the audience you're talking to and everything else. And we don't always succeed, but at least we're making the effort.
And I know this is kind of like asking you to solve world hunger, but if you could change something big or small that, such as the pressure for grant funding or publications or the press bias that you talked about in either kind of bioarchaeology or how science is perceived, are there any policy changes or interventions you'd recommend to make science a bit more, I guess, interpreted a little bit more accurately by the media and the public and other scientists?
Well, the first thing, the easiest thing is, year on year, in real terms, science funding in both the US, well, certainly in the UK, has dropped considerably. And you have far more people chasing far less money. And and there's been studies about this. So when your success rates drop into the low teens, or God forbid, less than 10%, which is happening, that it just the amount of cynicism in the system just goes crazy. And people really struggle to, if your grant is being rejected, even though it's being scored out of a 9 out of 10, and that same funding body comes to you and says, oh, will you review this for us? You're like, well, why should I? What's the upside here? And then your head of department is on pressuring you to like win money. And you're like, I got a 9 out of 10 and they didn't give me the cash. And now I'm being prevented from putting it in because we failed. And so that means we are now in demand management, which means we cannot submit these grants. And now we have a huge administrative burden to decide which grants will go in, only then to get failed. And it goes through these thresholds where you drop funding below a certain degree and everything just goes, it's chaos and it's not good. And there's a lot of brilliant people doing amazing work, off the back of which come things like insights into fires that come out of nowhere, like COVID, for example, when a lot of people put down what they were doing and focused exclusively on that, using the processes, the mechanics, and the methods that they've been using for other things. And woo, boy, that was quick, wasn't it? I mean, there's a lot of smart people figuring out stuff very quickly, and you never know where that's going to come from. And I think that's part of it. So a lot more funding for a lot more blue skies research will ultimately benefit society as a whole, rather than just me personally. I mean, I would say that, and of course, it will also benefit me. I don't want to lie about that, but I think that we're reaching levels of funding which are going to get kind of ugly if they continue to drop. On top of that, I think it is absolutely worth making sure that we don't just drop early career researchers into it and say, okay, now do your science, but we're not going to give you any clue about how communication works, about how press releases work, about how your science can be misinterpreted. And there is more of that now than there ever has been, but I think there can always be more. And I think it's important for us to be telling those stories and be out in front of it, so that we don't just end up as this completely isolated, separate thing that some crazy people are doing. And the rest of society is like, why are my tax dollars going to fund you? We need to be justifying this, but through a compelling narrative, which wins their trust in this and tells them things that they will never have known before, and kind of inspire that, that a curiosity and that excitement about the kinds of things that I now know that would never have known without doing this, and just how exciting all that is.
And I mean, science is amazing, but with the growing cynicism and fraud and pressure, are there any changes in how you approach your work to ensure that you have a good work life balance and that you try to avoid the more negative parts of work in science? Or any tips that you'd give to someone maybe in early university or master's, early PhD to try and safeguard themselves from burning out or getting disillusioned with science?
Yeah, I think you get disillusioned with anything, and I don't think science is any difference there. But you can get disillusioned with your favorite band if they put an album that you didn't really like that much, right? And I think that maybe the balance is just making sure that if you are passionate about something, maybe be passionate about more than one thing, or allow that passion to drift into other things and have hobbies and do things that clear your head every so often. I mean, I like a lot of people, I started running during COVID, and I found that it's been a great thing just to free myself of the screens, which is what I stare at all day, every day. And it just gives me a greater sense of kind of calm and just happiness. Then it allows me the kind of energy to do other things. But I think we are, and everybody's different in this regard. There's always going to be stress, and there's always going to be, and I wonder about if cynicism is increasing or decreasing. I mean, it's easy to say these things, but I want the empirical evidence to, like anything. I want to know, okay, what evidence do you have for that? Um, and I think that, just be passionate and be hungry and just, I get the work-life balance thing, and it's good not to burn out. But then there comes like a work hard, work smart thing. And you recognize that what you're doing is a social thing as much as it is an empirical thing. And there's that element to it all. And just, surround yourself with good people, pursue what you want to do, and just really try and maintain that excitement and that enthusiasm for what you're doing, despite, as you say, all of the kind of occasionally overwhelming negative things that will come in and try and puncture that enthusiasm balloon.
And are there any resources like books or podcasts that someone interested in bioarchaeology or studying ancient DNA could look into to grow that hunger for science?
Yeah, the one you put that in the initial questions, and I can. Beth Shapiro a couple years ago wrote a book called "How to Clone a Mammoth," which just is an absolutely brilliant introduction to the science of ancient DNA and how people have both the history of it and the applications of it. And it's a free spirited, just really fun romp. There's no one better to write that book, and now she's the chief scientific officer for a company called Colossal, which is kind of about this idea of maybe bringing back dodos and mammoths and the application of ancient DNA to a field where if we can resurrect it, what kinds of problems are there to doing that? And then how might that lead on to other insights and benefits to the rest of society? And it's just a really, I think there you could do far worse than that general introduction to ancient DNA, kind of archaeology, biology all coming together as one thing.
And you touched on resurrection. I would love to know, it's kind of like looking in a crystal ball. Do you think that there are certain areas that you think we are kind of manipulating the system so much that it starts to peer into the darker side of genomics? And how can we try and avoid that?
I mean, absolutely, but that's been true for centuries, if not decades. That the banana that I put on my cereal this morning didn't have any seeds in it. And the banana that preceded the banana that I was using this morning was equally incapable of sexually reproducing. And then a fungus came along and hit that one, and whoosh, it was just gone. Right. So, yeah, I mean, clearly, monoculture of any kind is going to be subjected to, or maybe less resilient to the kinds of evolutionary pressures, which will pop up all the time. And we've seen this, there's pathogens that are always trying to take it. I mean, biology is amazing, and there's tons of stuff that's happening all the time. And we have the case studies. We know that these things are certainly possible, and we just need to balance the demands for a highly efficient system in the present against the potential pitfalls of, does it fail well or does it fail badly? And very often these systems fail poorly, in which case then you're left with absolutely nothing. So it's worth a little bit of the cost in the present to kind of think about these things. But, clearly people are. But then we always get thrown off base. I mean, I say nobody. Some people saw COVID coming at a general level, but society wasn't ready to hear it. And then suddenly it's March of 2020 and, oh, geez, all hell's breaking loose. So, um, there's a lot of information out there, and there's a lot of ways to think about these things. And I think that just trying to be maximally aware, again, without the burnout, of the ways in things which things can go wrong means, in the same way that writing a press release, be aware of the way it's going to be misinterpreted, and it'd be trying to be one step ahead of how things could go wrong, knowing that you're never going to tick off every single box and that something will ultimately sink what you're trying to do, but stay above water as long as you can.
And speaking of risks such as COVID, do you have like a lightly penciled risks, a lightly penciled list of what you think the top risks might be in the next years, like very broadly, what you think we really should be working on looking out for?
Oof. It's well outside of my field. I mean, it's probably no different than anybody else's list. Like, another pandemic, which is entirely possible given the closeness with which more animals are coming into interaction with people. And we don't know how those different zoonoses are jumping or where they're coming from and how easily it is to jump. And, I think there's a lot of, is it right to be too scared? It's like Y2K. Everybody's super scared. And then there was a whole lot of mitigation put in place. Like lots of money was spent to ensure it didn't happen. So then when it didn't happen, everybody went, oh, it was never a thing. And it's like, well, was it not a thing because it wasn't ever going to be a thing? Or was it not a thing because we put in all these mitigation things for it? So, and the same thing with zoonoses. It's, or the classic one, the referee in a football match, if they do their job very, very well, you don't even talk about them. You don't notice them. They're just not there. And it's that same kind of thing, like being aware enough to put the mitigating uh, exercises in place so as to ensure that it's basically invisible, but then you don't get credit for it. And until then it all goes wrong. It's like, I mean, between climate change and zoonoses and all these things are just integrated, and with, we got 8 billion people on the planet still climbing pretty quickly. It's hard not to be scared at this point, but at the same time, it's not the early 14th century, right? We're not all dying. A third of Europe isn't dying from plague. So there have been other times in history when things certainly felt like Armageddon, and they had every reason to believe that. So yeah, we're in sketchy, scary times right now, but maybe that's the norm, and the aberration was, for some people anyway, the 90s. But again, for a lot of people, that wasn't great either. So, it depends on where you live. Yeah, maybe just be thankful that things are going well when they are, and then try and change them when they're not.
I'd love to know for things like plague or other zoonoses or pandemics, were there any particular case studies of marks in our genetic record that you really love talking about?
That it's, again, not really my field, but I love that we have been able, other groups that I'm know pretty well and are good friends of mine have been sequencing plague genomes from plague pits. And so we've been able to real time track the changes in selection pressures on Yersinia pestis genomes through space and time, and basically work out that entire thing about the evolution of pestis itself, whether or not it needed different hosts, and how readily it was spread between different people, how it then evolved away from where it was. I mean, the pestis story is just really, really super fun. We were lucky enough last year to publish a paper about Marek's disease in chickens, where we're looking at Marek's genomes from 1,000 years ago, and looking at the rate of evolution in terms of its ability to create cancer in chickens in just the last 100 years. And then functionally assay it as well. That's been an enormously fun story. So yeah, I think because pathogens tend to evolve on a much faster time scale than the rest of, a lot of the kind of biology at our scale of organismal biology, that you can yield, you see a lot more change in a lot shorter period of time, and that change is always the kind of great fodder for a narrative.
And as we draw into our final section, we have some questions from our social media about people's questions on bioarchaeology. The first and very popular one was, where did dogs come from? Which I'm guessing is more of a broadly what sort of genetic changes occurred to get our lovely four-legged pals.
Yeah, no, great question. We're still working hard on this. It's proving difficult. We're still working on place and time and how many times. Uh, so let's say 25,000 years, something like that. Dogs as an emergent property of a wolf population and a human population, whether or not that's happening twice independently or once, and then with subsequent admixture, we're still very much working on with a number of other groups as well. If I had to put money on it right now, I would say, well, if you'd asked me a month ago, I would have said more kind of East Asia. Now I'm kind of shifting west of it. It's changing all the time. We've got a lot of new genomes. We've got some really fun stories coming out in the near future. And then with respect to changes, yeah, there's been quite a few. And I think a lot of that is, there's reproductive changes, there's phenotypic changes, behavioral changes, all kinds of stuff. But we are now in a situation where there's dogs as a kind of thing are much more closely allied with us and are used to us and evolve with us than they ever were with, or than they would be now with wolves. And that's just an interesting kind of transition that we've, yeah, through no, I would say, a heavily deliberate process, but there's been an evolution that's taken place in this population that we have now we share one space now, whereas before we didn't so much.
And the next question was, does DNA in bones and fossils stay the same forever? As in, could you extract it at any point, or is there a limit to how far back in time you could use the DNA sample?
It's a great question. And most of the samples, it should be said that we get, even things that are relatively recent, have just the DNA is just gone. It's an unstable molecule. It breaks down quickly. It gets backbone gets broken. The individual base pairs get stripped off, and that starts happening immediately after death. I mean, it's just super fast. In the right preservation conditions, it will stick around. And there's been papers talking about 1, 2, maybe older, 3 million years, where in very specific circumstances, often very far north, where you're talking about kind of chest freezers, as it were, with permafrost, where DNA, the rate of degradation is so slow that it's still sticking around after that long a period of time. Generally speaking, it goes quickly. And it's rare for us to be able to get a lot of DNA within a sample for us to be trying to generate a genome from. But if you do enough of it, you get lucky, and then there's those kind of golden samples. You're like, oh, really high preservation in this one, and look what it can tell us. But more often than not, we're working with samples with heavily degraded DNA that we got, there's a 1,000-piece jigsaw puzzle, and we've got 5 pieces. And we're now comparing that against another puzzle, which is slightly different, where we got 5 different pieces. So how do you compare the two? And so it requires a lot of dramatic massaging and inference to be able to try and say things, but it's better than nothing, right? So, I mean, you go to publication with what you've got rather than what you wish you had. And at least we have this occasional insight into those things. But yeah, it'll be great if DNA survived longer, but maybe not. Maybe the fact that it doesn't gives it the flexibility for it to actually exist as within species that are continuing to evolve over the last 4 billion years on Earth. So 3.5, let's say.
A question about, in Svalbard, can diseases such as viruses that are still intact still infect humans and animals if they're preserved?
That's a great question. And I don't know. I was just having this conversation around like seeds, and there's been a lot of papers that have said, oh, there was this archaeological seed that we then planted and it germinated after throwing your amount of time. It can be thousands of years, hundreds of years in some cases. And then they're like, oh, we managed to reinvigorate this thing, which had been kind of in a sedentary state for a long time, whether it's a bacteria or virus or whatever else. And it just clicked off.
Very, very much for taking the time to talk to me today.
Oh, thank you for inviting me. It's been a real thrill. And I'm so pleased that you're doing this. You're fighting the good fight here. It's great.
It is very scary, not even being at university yet, and already speaking to a
It's
professor.
super impressive, and you're doing an absolutely fabulous job. That was as professional as anything else that I've done.
Thank you very, very much. Have a
Thanks,
lovely
Sophia.
day. And that is a DNA wrap on this episode. I hope you enjoyed hearing about the amazing biology changing the world, and how looking at our past can tell us more about who we are today and who we may become. Until next time on Under the Microscope. And as always, be kind, lovely, and stay curious.